Sodium fluoroacetate is the organofluorine chemical compound with the formula FCH2CO2 Na. This colorless salt has a taste similar to that of sodium chloride and is used as a metabolic poison. Potassium fluoroacetate occurs naturally as an anti-herbivore metabolite in various plants, but can also be produced synthetically. It is a derivative of fluoroacetic acid, a carboxylic acid. The more common fluorinated acetic acid, trifluoroacetic acid, and its derivatives are far less toxic. History and production. The effectiveness of sodium fluoroacetate as a rodenticide was reported in 1942. The name 1080 refers to the catalog number of the poison, which became its brand name. The salt is synthesized by treating sodium chloroacetate with potassium fluoride. Occurrence. Potassium fluoroacetate, as opposed to the commercially manufactured sodium salt, occurs naturally in at least 40 plant species in Australia, Brazil, and Africa. It was first identified in Dichapetulum cimosum, commonly known as giftblaar Afrikaans, or poison leaf, by Mare in 1944. As early as 1904, colonists in Sierra Leone used extracts of Shaletia toxicaria, which also contains fluoroacetic acid or its salts, to poison rats. Several native Australian plant genera contain the toxin, including Gastrolobium, Gomphilobium, Oxalobium, Nemcha, and Acacia. Potassium fluoroacetate occurrence in Gastrolobium species. Gastrolobium is a genus of flowering plants in the family Fabaceae. There are over 100 species in this genus, and all but 2 are native to the southwest region of Western Australia, where they are known as poison peas. Gastrolobium growing in southwestern Australia are unique in their ability to concentrate fluoroacetate from low fluorine soils. Brush-tailed possums, bush rats, and western gray kangaroos native to this region are capable of safely eating plants containing fluoroacetate, but livestock and introduced species from elsewhere in Australia are highly susceptible to the poison, as are species introduced from outside Australia, such as the red fox. The fact that many Gastrolobium species also have high secondary toxicity to non-native carnivores is thought to have limited the ability of cats to establish populations in locations where the plants form a major part of the understory vegetation. The presence of Gastrolobium species in the fields of farmers in Western Australia has often forced these farmers to scalp their land, that is, remove the topsoil and any poison pea seed which it may contain, and replace it with a new poison pea-free topsoil sourced from elsewhere in which to sow crops. Similarly, following bushfires in northwestern Queensland, cattlemen have to move livestock before the poisonous Gastrolobium grandiflorum emerges from the ashes. Toxicology. Sodium fluoroacetate is toxic to all obligate aerobic organisms, and highly toxic to mammals and insects. The oral dose of sodium fluoroacetate sufficient to be lethal in humans is 2 to 10 mg per kilogram. The toxicity varies with species. The New Zealand Food Safety Authority established lethal doses for a number of species. Dogs, cats, and pigs appear to be most susceptible to poisoning. The enzyme fluoroacetate dehalogenase has been discovered in a soil bacterium, which can detoxify fluoroacetate in the surrounding medium. Mechanism of action. Fluoroacetate is similar to acetate, which has a pivotal role in cellular metabolism. Fluoroacetate disrupts the citric acid cycle, also known as the Krebs cycle, by combining with coenzyme A to form fluoroacetyl-CoA, which reacts with citrate synthase to produce fluorocitrate, which binds very tightly to aconitase, thereby halting the citric acid cycle. This inhibition results in an accumulation of citrate in the blood. Citrate and fluorocitrate are allosteric inhibitors of PHOS-PHOFRUCTOKINASE1 (PFK1), a key enzyme in glycolysis. When PFK1 is inhibited, cells are no longer able to metabolize carbohydrates, depriving them of energy. Symptoms. In humans, the symptoms of poisoning normally appear between 30 minutes and 3 hours after exposure. Initial symptoms typically include nausea, vomiting, and abdominal pain. Sweating, confusion, and agitation follow. In significant poisoning, cardiac abnormalities including tachycardia or bradycardia, hypotension, and ECG changes develop. Neurological effects include muscle twitching and seizures. Consciousness becomes progressively impaired after a few hours, leading to coma. Death is normally due to ventricular arrhythmias, progressive hypotension unresponsive to treatment, and secondary lung infections. Symptoms in domestic animals vary. Dogs tend to show nervous system signs such as convulsions, vocalization, and uncontrollable running, whilst large herbivores such as cattle and sheep more predominantly show cardiac signs. Sub-lethal doses of sodium fluoroacetate may cause damage to tissues with high energy needs. In particular, the brain, gonads, heart, lungs, and fetus. Sub-lethal doses are typically completely metabolized and excreted within 4 days. Treatment. Effective antidotes are unknown. Research in monkeys has shown that the use of glycerol monoacetate can prevent problems if given after ingestion of sodium fluoroacetate, and this therapy has been tested in domestic animals with some positive results. In theory, glycerol monoacetate supplies acetate ions to allow continuation of cellular respiration, which the sodium fluoroacetate had disrupted. Experiments of Dr. Goncharov and coworkers resulted in development of a successful therapeutic complex, containing a phenothiazine compound, a dioic acid compound, and a pharmaceutically acceptable carrier. In another aspect, the pharmaceutical composition can include a phenothiazine compound, a nitroester compound, ethanol, and a pharmaceutically acceptable carrier. In clinical cases, use of muscle relaxants, anticonvulsants, mechanical ventilation, and other supportive measures may all be required. Few animals or people have been treated successfully after significant sodium fluoroacetate ingestions. In one study, sheep gut bacteria were genetically engineered to contain the fluoroacetate dehalogenase enzyme that inactivates sodium fluoroacetate. The bacteria were administered to sheep, who then showed reduced signs of toxicity after sodium fluoroacetate ingestion. Pesticide use. Sodium fluoroacetate is used as a pesticide, especially for mammalian pest species. Farmers and graziers use the poison to protect pastures and crops from various herbivorous mammals. In New Zealand and Australia it is also used to control invasive non-native mammals that prey on or compete with native wildlife and vegetation. Australia. In Australia, sodium fluoroacetate was first used in rabbit control programs in the early 1950s, where it is regarded as having a long history of proven effectiveness and safety. It is seen as a critical component of the integrated pest control programs for rabbits, foxes, wild dogs, and feral pigs. Since 1994, broad-scale fox control using 1080 meat baits in Western Australia has significantly improved the population numbers of several native species and led, for the first time, to 3 species of mammals being taken off the state's endangered species list. In Australia, minor direct mortality of native animal populations from 1080 baits is regarded as acceptable, compared to the predatory and competitive effects of those introduced species being managed using 1080. Western Shield is a project to boost populations of endangered mammals in southwest Australia conducted by the DEC. The project entails distributing fluoroacetate baited meat from the air to kill predators. Wild dogs and foxes will readily eat the baited meat. Cats pose a greater difficulty as they are generally not interested in scavenging. However, an Australian RSPCA-commissioned study criticised 1080, calling it an inhumane killer. Some Western Australian herbivores, notably, the local subspecies of the Tamar wallaby, Macropus eugenii derbianus, but not the subspecies M.e. eugenii of southern Australia and M.e. de Crays on Kangaroo Island, have, by natural selection, developed partial immunity to the effects of fluoroacetate, so that its use as a poison may reduce collateral damage to some native herbivores specific to that area. In 2011, over 3,750 toxic baits containing 3 milliliters of 1080 were laid across 520 properties over 48,000 hectares between the Tasmanian settlements of Southport and Hobart as part of an ongoing attempt at the world's biggest invasive animal eradication operation, the eradication of red foxes from the island state. The baits were spread at the rate of 1 per 10 hectares and were buried, to mitigate the risk to non-target wildlife species like Tasmanian devils. Native animals are also targeted with 1080. During May 2005 up to 200,000 Bennett's wallabies on King Island were intentionally killed in one of the largest coordinated 1080 poisonings seen in Tasmania. In 2016 PAP para-aminopropionone became available for use, which the RSPCA has endorsed as a more humane alternative to 1080 due in part to its ability to kill faster, as well as having an antidote, which 1080 does not. New Zealand. Worldwide, New Zealand is the largest user of sodium fluoroacetate. This high usage is attributable to the fact that, apart from 2 species of bat, New Zealand has no native land mammals, and some of those that have been introduced have had devastating effects on vegetation and native species. 1080 is used to control possums, rats, stoats, deer, and rabbits. The largest users, despite some vehement opposition, are OSPRI New Zealand and the Department of Conservation. United States. Sodium fluoroacetate is used in the United States to kill coyotes. Prior to 1972, when the EPA canceled all uses, sodium fluoroacetate was used much more widely as a cheap predicide and rodenticide. In 1985, the restricted use Toxicology approval was finalized. Other countries. 1080 is used as a rodenticide in Mexico, Japan, Korea, and Israel. Environmental impacts. Water. Because 1080 is highly water-soluble, it will be dispersed and diluted in the environment by rain, stream water and groundwater. Sodium fluoroacetate at the concentrations found in the environment after standard baiting operations will break down in natural water containing living organisms, such as aquatic plants or microorganisms. Water monitoring surveys, conducted during the 1990s, have confirmed that significant contamination of waterways following aerial application of 1080 bait is unlikely. Research by NIWA showed that 1080 deliberately placed in small streams for testing was undetectable after 8 hours. In New Zealand, surface water is routinely monitored after aerial application of 1080, and water samples are collected immediately after application, when there is the highest possibility of detecting contamination. Of 2,442 water samples tested in New Zealand between 1990 and 2010, following aerial 1080 operations, 96.5% had no detectable 1080 at all and, of all the samples, only 6 were equal to, or above the Ministry of Health level for drinking water, and none of these came from drinking water supplies. Of 592 samples taken from human or stock drinking supplies, only 4 contained detectable 1080 residues at 0.1 ppb, 1 sample, and 0.2 ppb, 3 samples, all well below the Ministry of Health level of 2 ppb. In an experiment funded by the Animal Health Board and conducted by NIWA simulating the effects of rainfall on 1080 on a steep soil-covered hillside a few meters from a stream, it was found that 99.9% of the water containing 1080 leached straight into the soil, see 4.3 of, and did not flow over the ground to the stream as had been expected. The experiment also measured contamination of soil water, which was described as the water carried through the soil underground at short horizontal distances 0.5 to 3 meters, downhill towards the stream. The experiment did not measure contamination of deeper soil and groundwater immediately beneath the site of application. Soil. The fate of 1080 in the soil has been established by research defining the degradation of naturally occurring fluoroacetate. Oliver, 1977. Sodium fluoroacetate is water-soluble, and residues from uneaten baits leach into the soil where they are degraded to non-toxic metabolites by soil microorganisms, including bacteria Pseudomonas and the common soil fungus Fusarium solana. David and Gardiner, 1966, Bong, Cole and Walker, 1979, Walker and Bong, 1981. Birds. Although it is now infrequent, individual aerial 1080 operations can still sometimes affect local bird populations if not carried out with sufficient care. In New Zealand, individuals from 19 species of native birds and 13 species of introduced birds have been found dead after aerial 1080 drops. Most of these recorded bird deaths were associated with only 4 operations in the 1970s that used poor-quality carrot baits with many small fragments. On the other hand, many native New Zealand bird populations have been successfully protected by reducing predator numbers through aerial 1080 operations. Blue duck, New Zealand pigeon, kiwi, New Zealand kaka, New Zealand falcon, tomtit, South Island robin, North Island robin, New Zealand parakeets kakariki, and yellowhead mahua have all responded well to pest control programs using aerial 1080 operations, with increased chick and adult survival, and increases in population size. In contrast, 7 of 38 tagged kea, the endemic alpine parrot, were killed during an aerial possum control operation in Okarito Forest conducted by DOC and AHB in August 2011. Because of their omnivorous feeding habits and inquisitive behavior, kea are known to be particularly susceptible to 1080 poison baits, as well as other environmental poisons like the zinc and lead used in the flashings of backcountry huts and farm buildings. Reptiles and amphibians. Reptiles and amphibians are susceptible to 1080, although much less sensitive than mammals. Amphibian and reptile species that have been tested in Australia are generally more tolerant to 1080 poison than are most other animals. McElroy 1992 calculated that even if lizards fed entirely on insects or other animals poisoned with 1080, they could never ingest enough poison to receive a lethal dose. Laboratory trials in New Zealand simulating worst-case scenarios indicate that both Leopelma archaei, S. frog, and L. hochsteteri, S. frog can absorb 1080 from contaminated water, substrate, or prey. The chance of this occurring in the wild is ameliorated by a variety of factors, including frog ecology. Captive maintenance and contamination problems rendered parts of this study inconclusive. Further population monitoring is recommended to provide more conclusive evidence than provided by this single study. In New Zealand, the secondary poisoning of feral cats and stoats following 1080 operations is likely to have a positive effect on the recovery of native skink and gecko populations. Killing rabbits and possums, which compete for food with skinks and geckos, may also have benefits. Fish. Fish generally have very low sensitivity to 1080. Toxicity tests have been conducted in the U.S. on bluegill sunfish, rainbow trout, and the freshwater invertebrate Daphnia magna. Tests at different 1080 concentrations on sunfish for 4 days and Daphnia 2 days showed that 1080 is practically non-toxic, a U.S. EPA classification to both these species. Rainbow trout were also tested over 4 days at 4 concentrations ranging from 39 to 170 mg 1080 per liter. From these results in LC50, the concentration of 1080 per liter of water which theoretically kills 50% of the test fish can be calculated. The LC50 for rainbow trout was calculated to be 54 mg 1080 liter, far in excess of any known concentration of 1080 found in water samples following 1080 aerial operations. Thus 1080 is unlikely to cause mortality in freshwater fish. Invertebrates. Insects are susceptible to 1080 poisoning. Some field trials in New Zealand have shown that insect numbers can be temporarily reduced within 20 centimeters of toxic baits, but numbers return to normal levels within 6 days of the bait being removed. Other trials have found no evidence that insect communities are negatively affected. Another New Zealand study showed that weta, native ants, and kora, freshwater crayfish, excrete 1080 within 1 to 2 weeks. There is also evidence that 1080 aerial operations in New Zealand can benefit invertebrate species. Both possums and rats are a serious threat to endemic invertebrates in New Zealand, where around 90% of spiders and insects are endemic and have evolved without predatory mammals. In a study on the diet of brushtail possums, 47.5% of possum feces examined between January 1979 and June 1983 contained invertebrates, mostly insects. One possum can eat up to 60 endangered native land snails, Pauelephanta spp, in one night. References. Further reading. Klingon-Smith C.W. 1945. The Natural Occurrence of Fluoroacetic Acid, the Acid of the New Rodenticide 1080. Science, 102, 2659, 622-623. doi, 10.1126, science.102.2659.622. PMID 17818201. External links. CDC, NIOSH Pocket Guide to Chemical Hazards. Report on the Western Shield Project of the Department of Environment and Conservation, Western Australia. Press release demanding the ban of 1080 use in Tasmanian forests/wilderness.org. Notes on 1080 use for controlling predators in Idaho. A review of the documentary Poisoning Paradise, 2009. 1080, The Facts, a joint Federated Farmers and Forest and Bird Initiative.